Let's start the session. Welcome to the selected talk, session number 4, new methods. And as the field of single-cell genomics is rapidly growing, there are a growing number of interesting technologies and utilization of methods into various biological systems. And we look forward to many interesting presentations as well as discussions from the group. So each selected talks are 10 minutes long, and I will followed by 2 minutes of Q&A. Please use the Q&A button on the bottom to ask questions. I will give the speaker a 5-minute warning, 2-minute and 1-minute warnings by the chat. So please stay on time. So our first speaker of the session is Dr. Lijia Ma, and she will talk about Direct-seq, which enables high-content molecular phenotyping in CRISPR screens. So just to give a quick bio of Dr. Ma, she received her PhD in bioinformatics from Beijing Genomic Institute, Chinese Academy of Sciences in 2009. And she was appointed as the deputy head of the bioinformatics department of BGI Shenzhen between 2007 and 2009. Dr. Ma moved to the University of Chicago in 2010 as a postdoc scholar, then appointed as a staff scientist since 2014. And she'd been deeply involved in various projects, including NIH ENCODE, ModCode, functional characterization center projects. Currently, since 2018, she joined Westlake University and its functional genomics laboratory. And her particular interest is in regulatory events that involve a cell fate determination and transition. So thank you, Dr. Ma, for accepting this talk, and we look forward to your presentation.
Hello, hi everyone, can you hear me? Good afternoon, everyone. It's glad to be online to participate in the HCA Asia meeting. My name is Lijia Ma from the Westlake University. My lab is focusing on developing functional genomics technologies to study the design principle of the human genome and how they were decoded in a spatial and temporal specific manners precisely. Today, I am going to share a short story that we recently published. The work is about a tool which we named as Direct-seq, and the challenge of this tool aims to tackle is to enable high-content molecular phenotyping in a widely used CRISPR screening assay. So let's start with a short introduction of the common steps of the CRISPR screening. It's quite often that we'd like to find a mutation that is corresponding to a certain phenotype we are interested from our daily research. Additionally, it would be great if we could get intermediate information along with the genetic information transfer, specifically, for example, the RNA expression profile given the phenotype. Nowadays, the CRISPR-based genetic screening has been widely used to serve for this purpose. In brief, the first step is to use a pre-designed CRISPR screening library to perturb the genome of a cell population. And obviously, we will then get a cell population with heterogenic genotypes. And then we would like to apply a selection pressure to these cells. For example, the pressure could be like a selection of marker gene expression or growth advantage or drug resistance. Eventually, the genotype distribution of the cell population will be shifted from its original distribution to the endpoint distribution. And through the analysis of the distribution shift, we will be able to know which kind of genotype is actually underlying the phenotypic changes. However, the plan does not that perfect as we expected, because there are 2 major issues along with this. Number one is that if we zoom into the endpoint cell population, it will still somehow to be heterogeneity in terms of their phenotype and genotype at some extent. And number 2 is that from the genotype to phenotype, we basically ignored the entire intermediate steps of the central dogma. Therefore, in order to get more comprehensive information from the endpoint cell population, we'd like to have a better method, which basically could allow us to study them in both higher resolution and better to be at a single cell resolution, and also could allow us to analyze the molecular information inside of these cells. Actually, this sounds very quite familiar because there are available technologies to help us to achieve this purpose. The most straightforward solution would be to combining the pooled CRISPR screening assay with the single cell technology. Nowadays, the challenge we basically transferred the challenge from the original described one to how we could connect the pooled CRISPR screening with the single cell RNA-seq technique. Now let's think about how we could do this. This is a simplified scheme. Let's think if we use the currently available single cell RNA-seq platform to analyze the endpoint cell population of the CRISPR screening, it's quite easy to get gene expression profiles from thousands of cells. But we won't be able to know the gRNA in each of the cells, as the gRNA transcripts are from pol-2 transcription, which means they have no poly-A tails and could not be captured in the regular RNA-seq experiment based on poly-A enrichment. And we will have two options. To resolve this problem. One is that we could change the gRNA itself. For example, we link the gRNA to a barcode, and the barcode could be will be transcribed by pol3 and therefore attached with a poly-A tail. And second, we can change the gRNA as well as the reverse transcription together, which we called we could basically embed a barcode into the gRNA itself, and then put a barcode specific RT primer in the RT system. Actually, different groups have done very beautiful works previously to try both strategies. The representative method of the first strategy is the, for example, the perturb-seq, CRISPR-seq, crop-seq, etc. And the second method are basically the widely used commercial solution, which 10x Genomics company named as feature barcode. In our hands, we basically use the Direct-seq technology in our lab has recently developed to basically combine these two strategies together. But our strategy actually have higher capture efficiency, and the design is more user-friendly. Let me explain why is this. First of all, here is a brief graphic summary of the Direct-seq. The left panel basically showing the wild type gRNA structure, gRNA scaffold structure. And in the right panel, we call it a programmed gRNA scaffold. Typically, in the tetra loop and the loop 2 and the tail position of this gRNA structure, there are 3 locations according to the Cas9 gRNA 3D structure that allowed us to insert the embedded capture sequences. In our case, we actually inserted a capture sequence which we called AG mixed capture sequence. The entire length is 30 nucleotide with 8 leading A at the 5' and followed by G inserted with every 7A. There are two major challenges of there are two major advantages basically applying this AG mixed capture sequence. One is that this AG mixed capture sequence basically mimics the endogenous mRNA poly-A structure, which could be efficiently captured by the oligo DT RT primer without introducing additional reagents in the reaction, because any disturbing to the reverse transcription reaction is disfavored, as this is basically the most critical step in the single cell RNA-seq experiments. And second, why we introduce G into the embedded capture sequence? Because we noticed that a pure poly-A capture sequence will significantly eliminate the editing efficiency of GI itself. And actually, the AG mixed poly-A sequence also existed in the physiological conditions as well. The next couple of slides will briefly show how we prove the efficiency and usefulness of this AG mixed capture sequence in the CRISPR screening experiments. And we also will show a demonstration of the Direct-seq on the 10x3' RNA-seq experiment. So we firstly demonstrated that the AG mixed capture sequence maintain a similar editing efficiency when we inserted it into the 3 different positions of the GRNA scaffold. As we mentioned previously, it's the tail position, the tetraloop position, and the loop 2 position. Also, we tested a few other options of similar design with G inserted into different position of the poly-A and also at a different frequency. You can see that actually there are some potential good candidates could be further explored in similar experiments. And afterwards, we also tested the specificity of the program, the scaffold, by examining the non-VEGF off-targeting sites, and we also tested the efficiency of the programmed scaffold on other targets. And finally, we also tested the editing efficiency by combining tRNA together with the GRNA in the program scaffold. Actually, it's very important that we use the tRNA-GRNA combination to enable these experiments, because the tRNA actually provided a very specific primer binding site to enrich the GRNA specific transcripts in the single cell RNA-seq experiments. We will explain this in the later slide. In the demonstration experiment, we applied a canonical experiment to screen negative regulators to the T cell activation. Here is a brief workflow of this screening experiments. Basically, we introduced a lentivirus GRNA library into a Cas9 stably expressed circuit cell line and selected for the top 25% of CD69 expression cells, as well as the bottom 25% CD69 expression cells. At the end of the screening, we collected the cells of the bottom 25% CD69 expression cells and made 2 sequencing library basically. One is the regular 3' mRNA library as exactly documented in the 10x manual, and the second library, which we called index library, is a specific GRNA transcriptor library we designed. There one thing, as I mentioned previously, the very special step we introduced into the system is that we introduced a tRNA specific primer in the library preparation step of the index GRNA library, so that the GRNA specific transcript could be specifically enriched during the library prep step. From the result point, you can see that basically almost all the cell barcode captured in the mRNA library can also be captured in our index GRNA library. This very efficient index GRNA enrichment could also be very directly visualized from the fragment size analysis as well. And finally, from our data analysis, we also find that those CD69 low expression cells, which supposed to be quite similar in terms of their phenotype, actually shows quite different gene expression profiles when we examined their gene expression profiles at a single cell resolution. Because the Direct-seq could enable the GRNA capture without any specific requirement to the RT primer or the system, it's actually compatible with most of the single cell sequencing platforms, which use oligo-DT as reverse transcription primers in the RT reaction. Here are three schemes of if you are going to use the 10x single cell 3' kit, or if we are going to use the 10x single cell 5' kit, or SmartSeq or SmartSeq on the C1 platform, how you could use this Direct-seq to combine it into your experiment. Finally, as a quick recap. So the take-home message is that use an AG mix capture sequence to mimic the poly-A tail of the endogenous mRNA is a major advantage of the Direct-seq, and the index GRA could be captured in reverse transcription by poly-DT, oligo-DT primers efficiently, and the index GRA could be enriched in the next generation sequencing library, and the enriched PCR step is highly recommended. At the end, the Direct-seq, both the manuscript and a detailed protocol have been published as shown in the slides. And with all of this, I would like to thanks for the committee to invite me to give this talk and my group. Thanks.
Great, thank you, Dr. Ma, for very nice technology to detect guide RNA in a very specific manner, and see a lot of applications with that. Unfortunately, we have very limited time in this session. So what can be done is that if you can click on Q&A button in the bottom, you'll see the questions that have been posted during your presentation. So what you can do is you can actually type the answers directly on the chat, and everyone will have access to that response as well. So that'll be much appreciated.
That's great.
Great, thank you. Thank you once again.
Thanks.